Okay, everybody, welcome back. Second week of distance learning for evolution class. Looks like everybody was able to access the video last week, and I didn't hear of any problems. I had a few people have troubles with the quiz, and so I've made the quizzes so that you can take them twice if you need. Um, so if you run into any problems, just let me know. Okay, so this week we're working on Chapter 9, which deals primarily with the issue of species and how they arise, so the process of speciation. So we're going to talk a bit about and think critically about what exactly a species is and how do they originate, what forces, what biological forces contribute to the origin of new species. And if you look at the right, there's a poster of what some of the fish diversity looks like in a lake in eastern Africa, Lake Tanganyika. And you might notice here that this says Lake Tanganyika cichlids. You might have heard quite a bit about cichlids in your intro bio class, or if you've taken genetics, they are a model system, a pretty remarkable instance of evolution of diversity. And so you can see here that the cichlids of Lake Tanganyika are quite a diverse group, lots of different colors, lots of different shapes. And that's a pretty interesting scenario, in part because this lake has more than 250 species of cichlids, just cichlids, not other groups of fish, but cichlids. And they've evolved from very few ancestors, fewer than 16 species got into this lake, is our current estimation. And this happened less than 15,000 years ago. So, in a very short amount of time, we've gone from having very few species, somewhere in the neighborhood of 16 species, that have diversified into this incredibly high number of species that are quite distinct in their appearance, in their ecology, their behavior, shapes, all of these things. So this is a great example that we'll be talking about a good bit more. And here you can see Lake Tanganyika. So that blue blob in the middle is the shape of Lake Tanganyika, and it's not that large of a lake. It's about 12,000 square miles. So you might think, yeah, that's enormous. Okay, certainly it's larger than Sardis. But if you compare it to large lakes in the U.S., for example, the Great Lakes, there are more than they span more than 94,000 square miles. And you'll remember that I just mentioned that there are over 250 species of just cichlids in Lake Tanganyika. We're not talking other things like catfishes or minnows or any of those things. Just cichlids in Lake Tanganyika. Well, in all of the Great Lakes, there are only 175 species, and that's all the fishes. Minnows, bass type species, catfish, all of those things, there are only 175 species. So why are there so many species in Lake Tanganyika? Okay. In this image, you can see the species of Tropheus, which is a genus of cichlid that is found in Lake Tanganyika. And you can see that if you go around the lake and sample, let's say you go fishing and you catch some Tropheus right on the edge of the lake in any one of these areas, depending on where you are on the lakeshore, you're going to catch very different fish. Okay. In the northwest, you're going to get fish that might have a white or an orange or a yellow band around their mid body. Okay. Down in the south, you might get a fish that's orange with tiger stripes or black with tiger stripes. On the east coast, you might get a fish with a blue head and a yellow mid-body band. Okay, there's a great deal of variation in these fishes. Now, let me pose this question to you: if you went fishing around the Great Lakes and just walked miles upon miles along the lake shore and caught, let's say, crappie all around the lake shore, do you think those crappie would look this different from one another? I can tell you they wouldn't. Okay. And so there are really interesting dynamics that influence how species arise and what causes them to be different. And different environmental circumstances contribute to that. Okay. And if you look at all of these different fish in these different places around the shoreline of Lake Tanganyika, these Tropheus, what is it that makes these different species? Okay. If you were to catch them, if you were to catch a bright orange one with black tiger stripes on it, and then, a week later in a different area, catch a solid black fish with a bright white mid-body band, you might think, and correctly, that those are different species. But what is it that makes them different species? Is it just the fact that you can tell them apart? If you look here, you see a man, looks like he's in Central Park, feeding some pigeons, right? And you've likely seen these pigeons somewhere, if not here in Mississippi, maybe a larger city or a park. But if you look closely at these pigeons, you'll notice that you can identify a number of different forms of pigeons. Some of these are almost solid white. Some are a light gray with 2 black bands on their wings and a greenish area around their head. Some of them are largely black or dark gray without any light coloration. So I can tell all of these pigeon types apart. Does that mean they're different species? Look at these puppies, right? I can tell all of these puppies apart. Some of them are black, spotted on a white background. Others of them are solid tan, kind of like the mother here. So what is it that makes things different species? Is it if I can tell them apart? What really makes a species is that it forms its own distinct gene pool, okay? A group of individuals that are genetically similar, that interact with one another reproductively and exchange alleles, okay? That's really how we get down to things, okay? Another great example of the diversity of organisms, most of you, all of you probably have been to a beach before in your life and seen these small birds running around the shoreline. But even though you've seen these birds running around the shoreline of a beach or even a lake, you probably haven't looked closely at them enough to think about critically whether the things that you're seeing represent one species or multiple species. And in fact, if you look at the silhouettes at the bottom, you'll see that there are quite a few different species. The names are there, so you know they're different species. That these are all things that you might see running around the shoreline at the beach. You can see a dowitcher, a whimbrel, a yellowlegs, a phalarope, a godwit, a killdeer, sandpipers. All of these things live in the same area. But if you look at them, what really stands out in these silhouettes? There's a reason I chose these silhouettes. And it is because if you look at what these birds are doing along the shoreline, they're running around, and as the water recedes, they're shoving their beaks into the sand and eating little critters out of the sand. Okay? Now look at all the beaks in those silhouettes at the bottom. They are all very different beaks. Okay? Some of them, like the godwit and the whimbrel, have very long beaks. Others, like the phalarope, the killdeer, and the sandpiper, have much shorter beaks. Okay? So if you think about these different species living in the same place, what makes them different species? Well, one of the things that makes them different species and allows them to coexist in the same area is the fact that they are doing very different things. So first of all, they're not breeding with one another. Secondly, they are coexisting in the same place because they're doing very different things. They are accessing different resources. The dowitcher is eating invertebrates down in the sand that are much deeper in the sand than the red phalarope can access. Okay? So they're eating different critters. And this contributes to our ability to recognize them as different species. And in situations like this, it's very clear that these are, in fact, very different species. But I just gave you an example with those pigeons where just because something looks different doesn't necessarily mean that they are different species. The appearance of an organism can affect whether they interact reproductively, okay? So here you can see there's a chimpanzee dressed up in a dress sitting, that's not quite at a bar. It looks like it's a telephone switchboard. Okay. That is not an organism that appears to be a potential mate to any human, right? The same is true of the different populations of Tropheus in Lake Tanganyika. Okay. They may appear largely similar, okay, in body shape, and the things that they eat are probably similar. So this is the complete opposite of those birds, those shorebirds that I was just showing you. These things largely eat the same thing. They largely live in similar habitats, and they are about the same shape and size, okay? But their appearance, their coloration is dramatically different. And this does influence in these species whether they interact reproductively, okay? A female of one is not going to mistake the other species for a potential mate. And that's going to be the subject of our additional reading this week, which I'll get to at the end. And so really, when we are thinking about what is a species and what makes a species, it's all about whether reproduction occurs, whether sperm and egg come to be in the same place, and whether they form a viable offspring. Okay, so we're going to dig into all of the components of this. Okay, so does it work? What do I mean, does it work, right? Sperm and egg come together. Well, just because sperm and egg end up in the same place doesn't mean that the product of that interaction is going to result in a viable offspring. Here you can see 3 instances that illustrate to you, first of all, why people use the term breed like rabbits. But these are clearly instances where the result of this interaction is not going to be a viable offspring. Okay? So this brings us to, how do we define a species? Do we use whether or not a viable offspring is created from their sexual interaction of 2 different individuals? Is that how we define what a species is? So this brings us to the idea of what is a species. And we use species concepts to basically establish a rubric that we can use in order to determine whether 2 things are simply slightly different populations of the same species or whether they are in fact distinct species. So by far the most widely utilized species concept is the biological species concept. And this is the idea that species should be defined as groups of individuals or populations that either actually or potentially interbreed. Okay? And then they are reproductively isolated from other such groups that are interbreeding with one another. Okay? So here you can see a photograph of a peacock, not one of mine, displaying to 2 chickens. Okay? Now, these are 2 distinct species. I know that, and you know that, but what makes them different species? Based on the principle of the biological species concept, what makes them different species is the fact that that peacock is not going to mate with either one of those hens. They are not going to, together, produce viable offspring. They do not have the potential to reproduce with one another. Now, this gets a bit tricky in many cases, and we're going to go through some examples that highlight how difficult this is to establish and what factors may play into determining whether things actually or potentially interbreed with one another. Okay? So like this weekend, when I go around my property and flip some of the tin that I've laid out as snake traps, essentially. So if you don't like snakes, don't leave tin lying around your property. Give it to me so I can put more around my property. But this weekend, I went around and flipped these 2 pieces of tin that are about 15, 20 feet apart. And under one, I found the speckled king snake, that black and yellow spotted species. And under the other, I found a corn snake. Now, I know these to be distinct species. They look very different, right? But do they look any more different than, say, the pigeons that I showed you in Central Park? Okay, why are these different species? Well, they have very different morphologies, they have different ecologies, they eat different things. The king snake eats snakes, the corn snake eats rodents and lizards. Okay, and so these two species are not going to breed with one another. Even though right now is snake breeding season and they are only 15 to 20 feet apart, they are not viable mates for one another. Okay? And it is the lack of reproduction between these two that creates or makes them different biological species. Okay? They are not, as we saw on the last slide, actually or potentially interbreeding populations. And as such, they are reproductively isolated from one another. They do not get together and produce offspring. So, what is it that prevents organisms from reproducing with other individuals that are not the same species? We call that reproductive isolation. And what might lead to reproductive isolation? Well, there are a number of things that can lead to reproductive isolation. The first is physical separation of populations. Okay, so we're going to think about in these next few slides the barriers or mechanisms that prevent different species from reproducing with one another. Okay, so in this instance, these two different species of spotted owl, the Mexican spotted owl and the northern spotted owl, they occur in very different places and they do not ever interact with one another because they live hundreds of miles away from one another. Okay, so these two populations are allopatric from one another. They are physically separated from one another. So it's important you understand what that word means, allopatry. Try is one form of the word. Allopatric refers to these two populations are allopatric with respect to one another. Okay, so now we don't necessarily know that if we brought together a northern spotted owl and a Mexican spotted owl into the same place, that they would be incapable of reproducing. What we do know is that does not happen on its own, and they do not exchange genes or alleles. Okay? And so because of that, they are considered a distinct species. Another thing that could prevent different species from reproducing with one another is temporal isolation. Okay? So these species might not be around at the same time. They might not be in the same place at the same time, or they may reproduce at different times of year. This example is a figure from further in the text, further on in the text. We'll hit on this one towards the end of the chapter again, but it's a good example of populations that are temporally isolated from one another. So in figure A here, you can see Lord Howe Island, which is a very small island in the Pacific. And B and C show two different species of palm tree that live on this island. And they are both wind pollinated. So their pollen is blown by wind around, and that is how pollen gets to other individuals and pollinates them so that they can reproduce. And so it's kind of surprising that you would have two very closely related species of palm on the same island, okay, that don't reproduce with one another. Well, the reason that they are not reproducing with one another, if you look down at figure D there, you will see that despite the fact that these palms are sympatric, okay, that's the opposite of being allopatric. Sympatric means they live in the same place. Despite the fact that these palms live in the same place, they reproduce at very different times of the year, okay? And so you see that in the Kentia palm, the blue line illustrates when the male flowers are produced. So male flowers are produced early. They produce all this pollen. Pollen starts blowing around, and then female flowers are produced on female palm trees, and they receive pollen from males that have already blown their pollen into the wind. Note that the curly palm has not yet flowered at all. Okay, it's a few weeks more before the curly palm male and female flowers are produced and release their own pollen and receive their own pollen. So because they are producing flowers at different times of the year, weeks apart, they are incapable of pollinating one another. Okay, this keeps these two species reproductively isolated from one another. Now, another way in which species might be prevented from reproducing with one another is if their offspring have lower viability, lower fitness. Okay, so this may manifest itself in either offspring that are weaker, they have a lower survival rate, perhaps the male and the female get together, sperm and egg come to be in the same place, but offspring simply have lower survival, if any survival. Okay, and then another example illustrated here by the horse. If you cross a horse with a donkey, you get a mule. Okay, and in that scenario, you end up with a sterile hybrid. You've seen this example before, and this is due to horses and donkeys having different numbers of chromosomes. And so their offspring have an unbalanced number of chromosomes. They're incapable of undergoing successful meiosis, and so they are therefore sterile. All right. So the horse and donkey are distinct species because despite the fact their genes, their alleles can mix in these mule offspring, it's a dead end. Okay. You will never get a horse that has 5% donkey genome or a donkey that has 5% horse genome because there's never any crossing back of a mule with a donkey or a mule with a horse. The offspring of this reproductive cross have little to no fitness in that they cannot reproduce themselves. Okay, so this is another way in which reproductive isolation might manifest itself. Now, I don't want to give you the impression that for every pair of species that you might compare, that there's only one factor contributing to keeping them reproductively isolated from one another. In many instances, it's a suite of characteristics, traits, and situations that prevent them from reproducing with one another. In this case, this example provided by your text, we have two different species of Mimulus, a genus of flower, that are pollinated by different species. So Mimulus cardinalis is pollinated by hummingbirds, and Lewisii is pollinated by insects. Okay. Now I want you to think about how Lewisii pollinate one another. Okay. How does pollen get from one Lewisii to another? It's carried by a bee. Cardinalis, on the other hand, pollinates one another with this intermediary pollinator species, these hummingbirds, going from one plant to the other. As they consume nectar, they carry with them pollen from plant to plant to plant, okay, cross-pollinating different individuals. So how then would you expect a cardinalis to pollinate a Lewisii or vice versa? You'd have to have a hummingbird visit a Lewisii or a bee visit a cardinalis, and that simply doesn't happen, okay? And so if you think about, or it happens very rarely, if you think about the contributors to reproductive isolation, it might be reasonable to assume that the primary and only mechanism isolating these species is their pollinator. And that turns out not to be the case, because not only do they utilize different pollinators, but they also occur at very different elevations, okay? So if you compound the fidelity, the fidelity of hummingbirds for cardinalis and the fidelity of Lewisii for, bees for Lewisii, right? Hummingbirds only go to cardinalis, bees only go to Lewisii. Now, we won't say that's necessarily 100% of the time. Maybe 2% of hummingbirds will visit both, 2% of bees will visit both. So there is the possibility that they might cross-pollinate. But the fact that they also live at very different elevations, they are geographically separated from one another, this also contributes to preventing even those few instances where cross-pollination might be possible due to some a pollinator species that's not necessarily doing what it usually would or should. The combination of differences in elevation and where they occur, and the species that pollinate them, contributes dramatically to this reproductive isolation between these two species. You'll notice also that pollen precedence, hybrid germination, and hybrid fertility contribute, though they don't contribute all that much. Okay, so pollen precedence simply means that the pollen of a Lewisii is going to have greater success pollinating a Lewisii than a Cardinalis pollen would. So if you put both types of pollen on a Lewisii, it's the Lewisii pollen that is going to have the highest chance of actually fertilizing that flower, just because they are biologically, chemically, genetically a better fit. Okay, so just keep in mind that reproductive isolation might be driven by a number of circumstances, not just a single factor. Okay. And so when we think about the barriers to hybridization, the barriers that prevent two different populations from mating with one another, they can be classified as pre-mating barriers, so barriers that prevent mating from happening at all, and post-mating barriers. Okay, now post-mating barriers are broken down into post-mating prezygotic, post-mating postzygotic barriers. Okay, so I want you to be very familiar with this table and understand, if I give you an example, whether the example represents a pre-mating barrier, a post-mating prezygotic barrier, or a postzygotic barrier. Okay, and they're very self-explanatory, right? A pre-mating barrier would be the physical separation of those two different species of owl. That prevents them from interacting with one another. The different pollination times of those palm trees is a pre-mating barrier that prevents them from pollinating one another. Okay, a post-mating prezygotic barrier means exactly that, right? Think back to that slide I showed you of rabbits mating with lots of different species. They mated, but did a zygote form? No zygote formed. Okay, why is that? Is that because of mechanical isolation? Okay, the genitalia of those two different species they weren't compatible, they didn't fit. Was it copulatory isolation? Was it gametic isolation? Okay, the difference between mechanical isolation and gametic isolation is, in mechanical isolation, you see those amusing photos of a rabbit trying to mate with a chicken or a cat. Okay, but the reproductive organs did not interact in a way that put sperm and egg in the same place. That's mechanical isolation. Now, gametic isolation is an instance where, let's say, that rabbit was successful in inseminating that dog. Okay, the egg and sperm are in the same place, but biochemically, they do not interact. Fertilization does not take place. Okay, so these are isolating mechanisms that happen after mating. Mating has happened, okay, but a zygote is never formed. The final category here illustrated are postzygotic barriers, and these are ones that I covered in the slide in some cases with the horse, donkey, mule example. Okay, so it might be that hybrids have lower fitness. It might be due to inviability, sterility of the hybrids, or it might be due to ecological or behavioral effects in those hybrids. Okay. Okay, your text in Figure 9.9 has an enormous figure legend that goes through a number of examples of reproductive isolation of different species. And these are things that I think you should read through and try to understand the differences or the specific instances described for each of these species, because each of them illustrates a very unique way in which these populations or different species are reproductively isolated from one another. Okay? And so to this point, we've been speaking primarily of a single species concept, and that is the biological species concept. Now we're going to discuss another commonly utilized species concept, and that is the phylogenetic species concept. Okay? So, unlike the biological species concept, that essentially relies on whether or not two populations are reproducing with one another or have the potential to reproduce with one another and produce viable offspring. This species concept focuses instead on the product of the evolutionary process, okay? Mutation, divergence, differentiation of populations, and basically, can we identify and recognize two different groups, and these different groups represent different species. They have different evolutionary histories. They have different characteristics. Those characteristics may be phenotypic characteristics. They may be genotypic characteristics. It may be both. Okay. And there are instances where we have species that differ genetically, but they don't differ in any way that we can see, right? Phenotypically, they don't appear to differ from one another. But that doesn't necessarily mean that they ought to be considered the same species just because they look the same, okay? So I showed you instances where things look different, but they're still the same species. There are also instances where there are two clearly different species that look identical, and they don't necessarily behave in different ways. But if you look at them genetically, they are clearly derived from very distinct gene pools that are not mixing. They are not interacting with one another. They're not breeding with one another. They are not exchanging alleles. Okay? They simply have not differentiated phenotypically from one another. So despite the fact that there are many more species concepts than the two that we have touched on in this lecture, these are the two that I want you to become familiar with and understand and be able to differentiate. These are the two most commonly utilized. All right. Now, this entire discussion of what is a species should not make you think that it is very clear what is and is not a species, and that species are clearly defined in all instances and easily identifiable. Because the lines that you draw between one population and another are difficult to establish. Okay? This is particularly true when you have species that live in adjacent areas, okay? And the populations on either side of that demarcation, that boundary, that border, if you will, for the species, are very distinct. Phenotypically, behaviorally, genetically, you can differentiate them. But sometimes, where those two populations come together, they will still interact. They will still interbreed. And this is what we call a hybrid zone. Okay? So a hybrid is the product of breeding species A with species B. You end up with a hybrid. Okay? And this figure from your text shows what goes on in just such a hybrid zone in these fire-belly toads that occur throughout Europe. And so what's interesting is there are two species here, the yellow-bellied fire-belly toad and the red-bellied fire-belly toad, two different species, okay? And if we think about what it means to be a Bombina bombina, or what it means to be Bombina variegata, so these two different species, they are distinct, well-recognized species, and as such, they consist of a very distinct gene pool. Bombina bombina has its own unique allele frequencies and mutations. and so does Variegata. But what's interesting is where the two populations come together, we have this hybrid zone, and there are instances where individuals of different species will breed and produce offspring with one another. Okay, so in figures A and B to the left, okay, the allele frequency and morphological score graphs, we are looking at, on the x-axis, distance from the center of the cline. So basically, 0 on the x-axis is where the red and the yellow populations meet, okay, geographically. And if you were to walk in a line to the left 30 kilometers or to the right 40 kilometers from that point where they meet, that's going either left or right on the x-axis, okay? So if you started, let's say, 20 kilometers west of this hybrid zone, this area where the two populations meet, that's the leftmost red dots in A, okay? So you start walking east, and as you get further and further east, you would notice that for particular genes, allele frequencies change very dramatically. Okay, so a particular allele that is rare in the West becomes incredibly common as you approach and then cross that interaction zone, that hybrid zone, okay? So allele frequencies are very different in these species. To the west, this allele that we're looking at is very rare. To the east, it's very common, okay? But right in the middle there, you can see that it has some intermediate frequency, and this is due entirely to the fact that in that area, these two species are mating with one another and bringing with them their respective allele frequencies. In the bottom left graph, this is a morphological score. So basically, think about it as how orange are the frogs that you find 30 kilometers to the west. They're not very orange, right? They're pretty yellow. So those red dots, those blue dots are quite low on the left side of the graph. And as you walk west, that morphological score increases very dramatically right at that 0 kilometer mark, okay? You go from yellow frogs to very quickly very orange to red frogs, okay? And so this is a clear illustration that despite the fact we have two distinct species that are genetically distinguishable, right? You can tell them apart genetically, that's the top graph. You can tell them apart phenotypically. They are hybridizing in this middle area. They are exchanging some alleles, okay? But that doesn't move very far west. That doesn't move very far east. And what I mean by that, I mean the alleles, the coloration. It doesn't change, okay, as you get away from that hybrid zone. So these are very distinct species, despite the fact that they are interbreeding. So this should highlight to you the difficulty of putting all of your support behind something like the biological species concept, okay? Because yes, Variegata is a actually interbreeding population, and all the Bombina are an actually interbreeding population. But the two are also interbreeding with one another to some extent and exchanging alleles. And so the application of the biological species concept runs into problems like this, okay? That doesn't mean it's not an important or useful concept. I'm just highlighting to you some of the difficulties that we run into as we try to pigeonhole organisms as being species A or species B. Now that you know that two good species, distinct biological species, can defy that foundational principle of the biological species concept in that they interbreed with one another, I'd like to introduce you to the concept of introgression. And introgression is the spread of beneficial alleles from one species to another, okay? And your text describes this example with species of Heliconius butterflies, in which the raid and the postman phenotype, look at the bottom left. This phenotype or this description refers to what is the color of the hind wing. So the back wing of these butterflies is either rayed, those orange rays radiating out, or this postman pattern. I have no idea why they call it postman, but it's that yellow bar on the hind wing. Okay. What's interesting is that this rayed and postman pattern that is present in Heliconius melpomene, you can see those two species. There's two lineages down at the very bottom, members of the same species, but different subspecies. Don't worry about subspecies. Those two members of melpomene possess these different phenotypic traits, but those traits have also shown up in two other species. So Timaretta has this postman pattern on its hind wing, and Elevatus has the rayed pattern on its hind wing. I think the clearest example here is Elevatus. So compare Elevatus to all of the other species that it is closely related to in that clade on the top of the tree, to Pardenalis, to Heclae, to Athelia, to Pneumata. It looks very different from all of those. Okay, it has this coloration. It received this genetic architecture for this wing coloration from Heliconius melpomene. Okay, presumably there was an introgression event, a mating between an Elevatus and a melpomene that led to these new alleles for this rayed pattern on the hind wing getting into that lineage. And it turned out to be beneficial and has now become fixed in Elevatus. Okay, this is the movement of genes and alleles from one species to another. And when beneficial, those alleles can persist. Something you've learned about before. You should have, if you took 160 here, you did some additional reading on a particular allele that exists in humans that is quite common in humans of Tibetan ancestry. This allele allows them to breathe and transport oxygen very effectively at incredibly high altitudes, right? Tibet is a very high elevation nation. Its people live at elevations where oxygen concentrations are much lower. Environmental or atmospheric oxygen concentration is lower than we are used to. And so they are able to breathe because they have this rare allele that allows them to transport oxygen more efficiently at lower partial pressures of oxygen. Where did this allele come from? It turns out that this allele is derived from another species of hominid, okay, Denisovans. So it's a very interesting instance of introgression that has affected our species. So why don't species freely interbreed with other species all the time in order to get benefits? Well, it turns out that hybrids between species are frequently inviable, and that inviability is quite often due to genetic incompatibilities, okay? And this is a phenomena described by a couple of scientists, Dobzhansky and also Mueller. And this phenomena is then thus named the Dobzhansky-Mueller incompatibility. And this is a phenomena describing how an ancestral organism might diverge into two different species, okay, and these populations go on to evolve and become different species. And they have their own unique mutations. They become fixed for different alleles. So in this illustration to the right, population 1 becomes fixed for this new allele A2, and population 2 becomes fixed for a new allele at gene B, B2, okay? So population 2 has alleles, they're homozygous for A1, they're homozygous for B2. Population 1 is homozygous for A2 and B1. If they were to come together and breed and produce offspring, those offspring would necessarily be heterozygous for A and heterozygous for B. it may be that these allelic combinations of being heterozygous for A, being heterozygous for B, are maladaptive. They do not work well together. And these F1 hybrids would have then much lower fitness and therefore prevent further interaction and genetic exchange between these two species. These types of genetic incompatibilities are quite often the result of large-scale chromosomal rearrangements. And by this I mean it's not just one species has a C at a nucleotide site and another species has an A. The mutations that happen are usually much larger in scale and on the order of chromosomal rearrangement. Okay, big chunks of chromosomes get inverted or transposed to another chromosome, okay, making these individuals incompatible. Your text describes here an example of these shrews in which different populations of even the same, what have been considered the same species, have different organization of their chromosomes. So if you look at the Novosibirsk population on the left, okay, they have this O and G region are combined into a single chromosome. Okay, what you're looking at here are replicated chromosomes. I put one up in the top right. You're more familiar with looking at human chromosomes like this that have been replicated, held together at the centromere. What we're looking at in this figure from the text are replicated chromosomes. And you'll notice that O and G in the population on the left are not in the same arrangement as they are in the Tomsk population on the right. Okay, so think about how a hybrid, what a hybrid is going to receive from their parents. Okay, if they get half of the Novosibirsk genome and half of the Tomsk genome, okay, they're going to have two of everything. They're going to have two of O, two of G, two of K, two of I, two of H, and so on, right? But then I want you to think about how they undergo meiosis, okay, and the gametes they would produce, and how those gametes would be partitioned certain chromosomes. Okay, they could end up with 2 copies of O or 0 copies of G, depending on what they get from that meiotic event. The hybrid you can see here ends up with this long connected set of chromosomes that really throws off meiosis. And so it is these scales of genomic rearrangements and mutations that generally lead to this type of genetic incompatibility. Okay? All right. So how does this whole process get started? Speciation generally results when there's some initial restriction to the exchange of alleles between populations that have begun diverging. Okay, and this is necessary because if you have lots of interbreeding between populations, what it does is it prevents genetic differences from really establishing themselves between the two populations. You know, look at these spotted owls in the bottom image. Okay, if owls are flying from Arizona to California and California to Arizona and taking with them alleles, are these two populations going to become different genetically? No, if they're exchanging alleles, then they're going to be the same species. So something has to prevent this exchange of alleles. And we've talked about temporal restriction to allelic exchange in the palm example, spatial examples in the case of the owls, or ecological restriction or isolation of these populations in the case of these mimulus flowers and their pollinators. Now, some of these things can evolve a lot faster than others. The differences between owl populations that are isolated are going to evolve relatively slowly, right? Presumably there's not a lot of selection for these two owl populations to look different. There's not necessarily a lot of selection on them to have different calls, mate attraction calls. Okay. So these types of differences that accumulate over long distance randomly are probably going to be pretty slow. However, traits that are under strong selection, such as those involved in mate choice, so sexual selection, those traits may evolve incredibly rapidly, and they may drive very rapid speciation, okay? Because if you look at these two species of hummingbirds that we have here in the United States, so on the left are the ruby-throated hummingbirds that are just now starting to come back through our area in northern Mississippi and presumably wherever you are back home right now. There's another species in the U.S., Anna's hummingbird on the right. These are both males in breeding coloration. And if you were a female of one of these species, you are unlikely to choose the wrong mate simply because they are so clearly colored that you're unlikely to make a mistake. Okay? And these preferences can evolve very rapidly. And small differences in what females prefer or what males look like over very short distances can lead to very rapid evolution and very rapid speciation. I want to come back to this cichlid example, and I've zoomed in on the phenotypic variation of the southern portion of the lake. And I want you to look at what these are all males. Males are brightly colored. Females are generally not so brightly colored in this as in most species. So I want you to look at this example and think about how this diversity of cichlid species evolved. Okay? And think about how each of these populations was presumably founded by some individuals. And in those populations, the males had some average coloration and females had some average preference for what their males looked like. Now, that slight difference in female preference and male average in each of these populations could very rapidly lead to the situation that we see here, where you have populations that differ phenotypically quite dramatically. And in each of these places, the females like what they see and breed with males that they are familiar with. If a female was to swim a little ways upshore or downshore and find another population in which males were quite different from what she was used to, she's unlikely to be terribly interested in mating with any of those individuals. And for that reason, genetic exchange between these populations is very low. Females don't want to mate with males that don't look like what they're used to or the population from which they came. New males that come to a new area that's already inhabited by another species. Females in that area prefer one phenotype, and some strange, unusual looking guy male cichlid comes into that population. He's not going to be well received. He's not going to have great reproductive success because he's not what the females in that area choose. And this is how, with these isolated little patches of cichlid habitat along the shore of this long lake, we've ended up with hundreds of distinct species. Okay, so this brings me to a concept that illustrates very clearly and remarkably how selection strongly drives the maintenance of genetic boundaries between species. Okay? And I want you to keep in mind, as you think about this example, the consequences of hybridization, the fitness of hybrids, okay? And the fitness of individuals who make the decision to hybridize, okay? So think back to that cichlid example that we just looked at on the previous slide. If a female cichlid of one species mates with a male that looks very different from her own species, what are her offspring going to look like? are they going to be attractive to potential mates of either species, hers or the father's? No. So because she makes a poor decision in mate choice, her offspring have low fitness, meaning she has low fitness. So making that decision on her part results in very low fitness for her and her offspring. And so that is selection acting against individuals that make that decision. Here we're going to talk about reinforcement. Okay, this is the principle that, or a clear illustration of the strength of that selection and the implications of making decisions that would result in hybrid offspring of lower fitness. So reinforcement clearly demonstrates this principle in that we have increased strength of prezygotic isolation in areas where two different species come into contact, okay? And this increased strength of prezygotic isolation happens because hybrids have lower fitness. So here we've got a frog, a green-eyed tree frog in Australia, and there are two very distinct lineages. So I would call them distinct species, a northern species and a southern species, okay? And if you think about these two different populations, I want you to look at the map. So we zoom in, and then we've got an even further zoomed in. You can see the range of the northern lineage. You can see the range of the southern lineage, okay? And that little pink spot is an area that is inhabited by southern species, okay? But it's surrounded completely by populations of the northern species, okay? So what we've got here are two species, okay? We just have an isolated patch of southerners up there in the north. And here I've got it's not as stylized a figure, but you can see the phylogeny on the left. Northerners and southerners. N for northerners, the blue. S for southerners, the red in the tree. And if you look at those isolated southerners in the phylogenetic tree, they are clearly members of that southern lineage, okay? Now, if you look at the map on the right, it's kind of frustrating in that the letters they used are Q in all situations instead of N and S. But in any case, you can see the red Qs way up there in the north, okay? And all around them are blue Qs. So that illustrates the genetic relationships of these southerners that are isolated and surrounded by northerners further north, okay? In the south, to the south, these southern populations are all genetically contiguous, as are the northerners, okay? All right. Now let's think about what was demonstrated in this study, which I'm not going to make you read, but it's a fantastic illustration of reinforcement. Okay. So here's what's going on there. On the bottom, we now have a graph of the mating call and the duration and note rate and the pitch. So I want you to think about this. We've got the call of the northerners, the southerners, and then those isolated southerners, okay? We've got, if we compare the northerners and the southerners, they have different calls, right? If you look to the right, the pitch is not very different between northerners and southerners. But if you look at the duration and the note rate, okay, the northern and southerners have different duration and note rate. So just think of the northern frog mating call as burr, burr, right? Two long calls. Instead of the southerners having burr, 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 right? It's faster, it's shorter, okay? But they're the exact same pitch, okay? But if you were a northerner, you could differentiate a northern female. You could tell whether the frog that you hear calling is a southerner or a northerner, because those calls are quite different. Not in pitch, but in duration and note rate. But look at the call of that isolated southern population up in the north. That call is much finer, longer, many more notes, and at a much higher pitch. I'm not going to try to make that call. That would be hilarious, but not going to do it. But you get the idea. What we see in instances of reinforcement is that the population that is at greatest risk of mating with an individual of the wrong species, wasting its reproductive output, lowering its fitness and that of its offspring, those individuals have shifted their phenotype so that they are much less likely to make that mistake. Okay? So selection is reinforcing the boundaries, the genetic boundaries, the species boundaries of northerners and southerners in areas where they are at most risk of making that mistake. Okay? So prezygotic isolation is enhanced, strengthened, reinforced in these areas where hybrids are most likely to occur and hybrids have low fitness. Okay? Now here I want to touch upon a pretty unique situation that arises not infrequently in plants, certainly much less commonly in animals, and that is the possibility of instantaneous speciation that frequently happens when two different species hybridize. And what we see when this happens is that sometimes species will hybridize, right? They produce haploid gametes. Those two haploid gametes come together in the same place, but it turns out that their chromosomes are not compatible. They are not homologous to one another. And so what happens is, let's say in this example here, we have two different sunflower species, T. dubius and T. pratensis. Both of them have an N of 6, right? So they're diploid species. They produce gametes that have 6 chromosomes in them, and they have these really not terribly informative illustrations of the 6 chromosomes that would be in the gamete of each of these species. And when those two gametes come together, you end up with 6 of dubius, 6 of pratensis, but you don't have 2 of anything. Okay. Now, what plants are able to do that rarely, if ever, happens in animals is that those chromosomes in that zygote, so the 6 dubius chromosomes, the 6 pratensis chromosomes in that zygote, will themselves spontaneously double. Okay. And then you end up with a species like Mycelius that has an N of 12. Okay. So the diploid living plant is 2N equals 24. Okay. It has 12 dubious chromosomes. It has 12 pratensis chromosomes. When it undergoes meiosis in its gamete, it puts 12 chromosomes, 6 dubious chromosomes, 6 pratensis chromosomes. This immediately, as soon as this zygote doubles those chromosomes that it got from each parent and becomes, instead of 2N equals 12, it's a 2N equals 24 plant. As soon as that happens, that plant, that one zygote, that seed that is growing, is a different species. Think about it. It could never fertilize its parent or its sibling or its cousin from T. dubius or T. pratensis. Okay? This is because they have completely different chromosome numbers that are incompatible with one another. And so this is a really unique situation in which speciation can happen incredibly fast. Super cool. Okay, so we're going to dig into the geographic component of speciation here for a bit. And what this figure from your text illustrates are the 3 primary geographic modes of speciation. Now, by far, the most common form of speciation is allopatric speciation. Allopatric speciation arises when populations are separated from one another by a barrier, and that barrier prevents them from exchanging alleles. So in this case, it's a mountain chain separating two populations of flowering plants. Somehow we end up with plants on both sides. Okay. Now we'll get into the two different ways that might happen here shortly, dispersal or vicariance. But let's not focus on that right now. Right now, we're going to focus on the fact that those two populations on either side of that mountain are isolated from one another genetically and physically. That isolation allows them to evolve independently over time through the process of mutation, drift, and selection. They become different phenotypically, ecologically, reproductively, in many different ways, such that if some point later in time, so time is, see the arrow on the left, at the very bottom, somewhere later in time, even if those two populations move back and forth and interact with one another, they've become different from one another. They are no longer reproductively compatible. They are genetically distinct, and they are therefore distinct species. Now, the parapatric speciation that we see up here, this is two populations near one another that become different. They're basically adjacent to one another, and they become more and more different over time, such that they might later, at the end time in this figure, overlap completely and be co-distributed throughout their ranges, but be reproductively isolated from one another. Okay. And I'm going to give you an example of that in a little bit that hopefully helps make clear how that might happen. Okay. The palm trees on Lord Howe Island that we already talked about are somewhere in between this and sympatric speciation, though they're used as an example of sympatric speciation. Okay. Sympatric speciation is one in which the populations are never isolated from one another. They always live in the same place. They differentiate over time and become distinct species while they've lived in the same area the whole damn time. Okay, this is a very contentious topic, contentious issue, whether or not this actually happens. There are very few instances that are agreed to have been clearly demonstrated, and we'll talk about one here shortly. Okay. But the difference between parapatric and sympatric speciation is that the process of speciation happens either while they're separated, even though they're kind of adjacent to one another in parapatric speciation, or while they live in the same place, not really living in different places in sympatric speciation. In the end, they're all sympatric. All three of these, they are sympatric. They live in the same places. Okay. But it's how they got to the point of being different species, where they were throughout that process. Were they physically separated from one another as in allopatric speciation? Did they live adjacent to one another with low levels of gene flow between them? Perhaps like the fire-bellied toad example we used. Or were they sympatric, living in exactly the same places and just not reproducing with one another? Now, one term I want to introduce you to is clearly illustrated in these bottom two figures for allopatric and parapatric speciation. And this is the principle of secondary contact. Okay. Now, what has happened in these two species as they went from one to two species, as they speciated? They were physically separated, isolated from one another, did not have any physical or genetic contact for quite some time. And that physical separation is really important in allowing those populations to become more and more different from one another over time, such that upon secondary contact, where that red arrow is, they live in the same place, they're in the same areas, they could breed, they don't breed. They don't breed because they've become so different, whether it's ecologically, genetically, behaviorally. Whatever the reason is that they don't breed anymore, it took a period of isolation for that to evolve, for that to happen. And so this principle of secondary contact is one that can only happen between two species after they've had sufficient periods of isolation and were able to evolve enough differences that make them reproductively incompatible. Okay. Now, I alluded to this a few slides ago, but I want to make clear to you that there are two ways you can end up with allopatric populations. Two populations of the same species that live in different areas. And that physical isolation then allows them to evolve independently and become different species. The two mechanisms are vicariance and dispersal. Okay? Vicariance is illustrated in this upper portion of the figure, in which you have a continuously distributed species A that in some way is divided. It is split into two populations. In this case, there's a river that divides the two populations, or you can think of it as the right side being a chunk of the continent that breaks off and becomes an island. Right? In this scenario, you have what was a continuously distributed species now is two different things isolated from one another. Okay? And it's very important to realize why they use the notation in the middle top figure. Instead of being A on the left and something else on the right, you have A prime on the left and A double prime on the right. Okay, or whatever the hell A parentheses. I don't even know. A quotation mark. Because I want you to think about what has happened. That continuously distributed species A has been divided into two, okay? Now, are either of those populations on the left or the right, are either of those species A? Of course not. Species A was characterized by all of the diversity across its entire distribution. Just like when you look at a phylogeny and look at the common ancestor of two species, and I ask you what species was that common ancestor, right? I think we use this example with the human and chimp, okay? The common ancestor of a human and a chimpanzee, was it a human? No. Was it a chimpanzee? No. Okay? It was neither. And similarly, neither of these two populations on either side of this river is species A. They are both necessarily something different, okay? Because they have had a significant portion of the species A gene pool removed from them, okay, and isolated on the other side of that barrier. Now, over time, each of these will evolve independently and become a different, distinct species. So in this case, species B and C. Again, neither of those is species A. Now, contrast that with what happens in dispersal, okay? On the left, we have a continuously distributed species A that some individuals cross that barrier and colonize a new area, okay? They colonize a new patch of habitat on the other side of that barrier, that riverine barrier, and they are then, that new population is immediately distinct from species A. It does not have the entire genetic diversity of species A. So it's something different. And over time, it can become something quite different. Okay? So in both of these cases, we end up with new species. It's just, how did that physical isolation arise? Was it due to division of the distribution of the ancestral species, or was it through the dispersal and expansion of the range of this species? Okay. Now, this ties into the principle of founder effect speciation, okay, which I want you to think about. So you read the text, okay, and if I haven't made this clear already, reading the text in this period where we are going through working virtually, it's much more important than it was when we were interacting in class and you were able to ask questions, okay? Read this section on founder effect speciation. This is an instance in which dispersal plays a large role in the process of speciation, okay? Here we have an example of these birds, these kingfishers, that live on this large island of New Guinea, okay? and they have this characteristic coloration of dark blue wings, light blue head, light blue shoulders, bright orange beak, kind of bluish tail. And they live in all of these areas 1, 2, and 3 on the Big Island. Okay. But some individuals have colonized these smaller outlying islands. So Island 8, Island 7, Island 6. Okay. What you find on those islands are birds that look quite different from the mainland. So even though they're very close to, say, population 1, okay, population 1 is really far away from population 3, but they look the same. They're really far away from population 2, but they look the same. They're quite close to population 7, but look how different they appear. This is due to the effects of founder effect. We talked all about founder effect, okay? How different allele frequencies can arise in populations that are founded by few individuals. Not many individuals got to that island, number 7, okay? And they brought with them unusual allele frequencies that are not representative of population 1. They brought with them potentially preferences of females for this new attractive dark blue coloration. And for that reason, that population evolved to become completely dark blue with a solid white tail. Okay, they look very different from the main New Guinean populations. Okay, so these populations, through founder effect, the principle of founder effect, this low genetic diversity, are diverging not only because of genetic drift, right, this sorting that's associated with the founder effect, but also through selection, potentially through mate choice, through differential survival of individuals with these different colorations. Okay, so this is an important contribution of this principle that we've learned, the founder effect, on the geography of speciation. So the principle of physical isolation, allopatry, and its contribution to speciation is quite clear from evidence that we have. And it's even apparent within species. Okay, so this is an illustration of the sexual isolation of populations of one species of salamander. Okay, and so this species of salamander is widely distributed, and if you collect individuals across the range of its distribution, which spans here 250 kilometers, okay? So what is this showing me? It's showing me how sexually isolated two individuals are that I put together in the same container to see if they will mate, depending on how far apart I caught them. If I caught them 20 kilometers apart, their sexual isolation is very low, right? So they have a high probability, high chance of mating with one another and producing viable offspring. If the two individuals I put in a container to see if they will mate were collected 250 kilometers apart, okay, there's a much higher sexual isolation index, a measure of how difficult it is for them to reproduce, basically. It's much less likely that they will reproduce if they were from far apart than if they were near one another. Okay? A really interesting illustration of the importance of geographic isolation, physical isolation, allopatry of individual populations in driving the process of speciation is illustrated by this pretty famous graph that shows you how much gene flow certain species have between populations, and on the y-axis, the minimal island size allowing speciation. So first, let's think about the x-axis, okay? How much gene flow is there between populations of snails versus populations of bats and foxes, okay? Which of those, if you have two populations that are 5 kilometers apart, for which species are you going to have more gene flow between those populations, snails or bats? Well, certainly bats and butterflies and foxes, for that matter, are going to be much more capable of dispersing and carrying with them alleles from one population to another. For a snail to move from one population to another, that's a lot more difficult. It does not happen very much. So gene flow between populations is scaled here for all of these species, okay? Bats and foxes and these large winged butterflies disperse pretty far. Other things like lizards, snails, some smaller flowering plants, and even some small birds don't disperse all that far, okay? Now, the y-axis is really interesting here because what it is showing you is the minimal island size allowing speciation, okay? So if you have an island whose area is 50 square kilometers, okay? So on the y-axis, look at 50. And I take a bat and release a bat species on that island. Is that one bat species going to turn into two? Will it speciate? No. Okay. The minimal island size for a bat to speciate is well over 1,000 square kilometers, right? Why is that? Why can't a bat speciate on an island that is only 50 square kilometers? It's because they move too much. They have too much gene flow. You're not going to have allopatric populations of a bat on a 50 square kilometer island. Now, what about a snail? Can a snail speciate on an island that's 50 square kilometers? Hell yes, it can. It can speciate on an island that is less than 5 square kilometers because they don't have much gene flow. They don't move much. You can have isolated populations of a snail on an island that's only 5 square kilometers. You cannot have isolated populations, allopatric populations with no gene flow of a fox or a bat on an island that's only 5 kilometers. Okay, so this makes clear the importance of allopatric speciation. Now, this graph is not just illustrating this phenomena for a fox, a bat, a butterfly. It is basically, this figure is representative of every island on the planet. And it's not just representing one bat or one fox or one butterfly. It is representing all bats, all carnivorous mammals, all butterflies. No butterfly speciation has been documented on any island less than 200 square kilometers. No bat or carnivorous mammal speciation has been documented on an island less than 1,000 square kilometers. Okay? The smallest island that has had bird speciation on it is 50 square kilometers. Okay? So the fact that there are no islands on this planet where butterfly speciation has happened on that island that are less than 200 square kilometers is incredibly strong evidence for the importance of physical isolation of populations. Okay? All right. Now, this brings me to this example that we haven't yet dug into on sympatric speciation. Okay? So can speciation happen sympatrically? Your text makes it quite clear. This is a very contentious issue because it's difficult to demonstrate after the fact. Okay? This example that I used of two palms that flower at different times on the same island as a mechanism for temporal isolation, allowing two different species to maintain their genetic distinctiveness, it's actually a system in which it is believed that these two species evolved on this island together, these wind-pollinated palms. They live on the same island, although they inhabit slightly different soils. Okay? And it's believed that the different soils that they live on affect their flowering time. Their uptake of nutrients differs at different times of year, and so they flower at different times of year. And over time, the plants that grow on more calcareous soils have not pollinated those plants that grow on other soils, and so they become more and more different over time. So like this sympatric speciation schematic in the bottom left, they live in the same place. They simply have evolved over time to flower at different times, to live on different soils, despite the fact they're on a very small area. They're sympatric with one another. But read through this example, and I want you to understand how, I've just emphasized with the previous slide how important allopatry is, physical isolation of populations, to the principle of speciation. I want you to realize how rare and contentious this idea of sympatric speciation is. Okay. And lastly, to focus on making sure that you understand parapatric speciation, I think the best example is the one utilized by your text. And it focuses on something we've already talked about, and that is the evolution of these different colored lizards within the same species in this white sands area of New Mexico. Okay. And so you can see what the white sands look like on the left, what the dark soil right around those white sand areas looks like on the right. And what we would have here, because this white sand area is so recently developed, it's within the last 5,000 years that all these white sands have been set up, the lizards that inhabit this area have colonized the white sands and become adapted to this new environment. And as they've done so, you can imagine dark lizards that go to the white sand area and white lizards that go to the dark soiled areas. They don't do so well. And so selection has favored individuals that stay in their areas. Their fitness is higher if they reproduce with individuals in their same areas with their same coloration. And what we've got below the images of the sand soil are two different structure plots for Holbroochia maculata and Scoloporus undulatus. Okay, so I want to make sure that you understand what these structure plots are showing. So each of these vertical bars, whether it be blue or green or both, are illustrating a single individual. So every one of those bars is an individual lizard. On the left side are individual lizards that were collected in the white sands. On the right side are individual lizards that were collected on the dark soil. They have the same number of Holbroochia and Scoloporus from the white sand. They have the same number of Holbroochia and Scoloporus from the dark soil, right? So that's why there's the same number of vertical bars. Or actually, it looks like there's more Scoloporus than there are Holbroochia because the bars are thicker for Holbroochia, right? So there's more vertical bars crammed in for Scoloporus. My bad. I just looked more closely at that. Okay, does that make sense? The bars represent individuals. Now, the color of the bar represents the proportion of the genome that is derived from a white sand ancestor, green, or the proportion of the genome that is derived from a dark soil ancestor, blue. Okay, so if you look at the Holbroochia collected from the white sands region, most of their genome is derived from a white sands ancestor. And conversely, for the dark soil populations, they're almost all dark soil ancestors. Now, it's not so clear with Scoloporus, right? You see a lot of Scoloporus in the white sands that have a big chunk of their genome from the dark soil. There's a lot of blue there. There's a lot of green on the dark soil side for Scoloporus, okay? What this tells me is that Holbroochia has been adapting to this difference. This speciation is much further along. So make no mistake, currently these are considered the same species, but there is clearly much less genetic exchange between Holbroochia populations than between Scoloporus populations. And that tells me Holbroochia is much further along in this process. Okay, so if you look over to the left, the schematic, they are maybe the third illustration in the schematic where it's purple on one side and yellow on the other, right? They are clearly establishing genetic differences across this environmental boundary. Whereas the Scoloporus are much more mixed. It's much more like the second from the top parapatric schematic, where you have purple flowers on one side and then orange and yellow on the other. They haven't quite established such a clear genetic distinction between the populations over time, but it's happening, okay? And over time, selection is going to continue to reinforce these genetic divisions between these populations. And eventually this will lead to speciation. Okay, this last slide is just touching on the topic of the reading that I've assigned for this week. And this reading focuses on the selection and the genetic basis of differentiation between rainbow darters and orange throat darters. So darters are these small fishes that live in the creeks and streams around here. They're incredibly diverse in this region. We've got lots of darters, even in the Yacna and the Tallahatchie drainages. Okay, the orange throat darters live in the areas denoted by the orange patches, and the rainbow darters by the blue. And the information you're going to be reading is based on a study that was recently published that looked at, if these two fish live in the same creeks and streams, why don't they breed with one another? Or do they? Okay, so I encourage you to go through that. I have made a few notes and questions within the document that you do not need to answer. They're there to help you understand the topic that you've just read about. Okay, so to be clear, you don't need to send me answers to those questions there. They're just there to make sure that you think critically about what you've just read. Okay? All right, that's it. More next week.